In continuation of the proteomics function, next topic is protein separation and purification in proteome analysis. So types of the proteomics, there is a functional proteomics, structural proteomics and differential proteomics. Proteomics is mainly involved in the identification and analysis of entire protein content. So functional proteomics is identification of protein-protein interaction, protein DNA and protein RNA interaction, they affecting the function. Structural proteomics, identification of all interactions by metal ion, toxins, drugs, etc. They affecting the protein structure. Differential proteomics, determination of the differences in the protein expression. So this is the overview of the proteomics. So proteomics expression, how the expressed in at cellular level, 3D structure of the protein, interaction. So these all can be studied by using the bioinformatics tools. So in bioinformatics, we can predict the 3D structure of the protein. So techniques involved in the proteomic study, how we can isolate the protein, how you can study the complete structure of the protein. So there are different techniques to identify the protein structure and their biological importance during the drug discovery processes. Some of the very basic analytical techniques are used as a major proteomic tool for studying the proteome of the organism. This is protein identification through different techniques. One is 2D gel, another one is MALDI TOF, protein array, proximity ligation, lab on chips, that is also called as a microarray chips. So steps in proteomic analysis. The following steps are involved in analysis of the proteome of an organism. First is purification of the protein. After purification, separation of the single protein. After separation, identification of the protein. After identifying the particular protein, next analysis of the protein molecule. First coming to the purification of proteins. This step is mainly involves extraction of the protein sample from the whole cell. Whole cell, tissue or a subcellular organelle, followed by the purification using the density gradient centrifugation, chromatography techniques, mainly exclusion and affinity chromatography. So next, after extraction of the protein by using the specific techniques, so next one is we isolated the group of the protein. From that protein we have to separate the single protein. So that is possible by using the 2D gel electrophoresis. It is applied for the separation of the proteins on the basis of their isoelectric point in one dimension and molecular weight on other dimension. So spots which are obtained by separation of the individual protein by the 2D gel electrophoresis, they are detected using the fluorescent dyes or radioactive probes. After separation of the single individual protein by using the 2D gel electrophoresis, next step is identification of the protein. Which protein being identified? The separated protein spot in a gel are excised and digested in a gel by the proteases, that is the protein digestive enzymes, for example trypsin. Diluted peptides are identified using the mass spectrum. In identification of the protein process, we identify the single protein or a protein that may be peptide or a polypeptide by using the different proteases, protein digestive enzymes called the proteases. So after obtaining the single peptide, the peptide mass is identified by using the mass spectrometry. That is analysis of the protein molecule usually carried out by MALDI TOF. MALDI TOF is expanded as a matrix assisted laser desorption ionization time of flight based peptide mass fingerprint. By peptide mass fingerprint, we can identify the single protein. So this is determined amino acid sequence is finally compared with available database to validate the protein. After identify the single polypeptide by using the multi-TOF, we can determine the peptide structure and name of the amino acid by using the database. Database is already stored in the different type of the amino acid sequences. So for that we have to select the online tools. By using the online tool, we can analyze the proteomics. That online tools are the Mascot, Aldinate, Popitom, QuickMod, Peptide Cutter. So by using this online tool, we can identify the single amino acid sequence of the identified peptide. So this is the overall process of the identification of the protein. Sample means the sample where you have obtained either from the whole tissue or from the cell or from the membrane, intracellular, extracellular, whatever the protein you have to isolate, that is called the sample. The sample is applied for the to be applied to the 2D gel electrophores. This sample is separated as individual protein sample as like a dot. Again, these dots are indicated the unidentified protein. This unidentified protein are extracted by using the gel. This is unidentified protein. This protein is digested by the proteases. After digesting with the proteases, it applies small peptides. The peptides are applied to the mass spectrum to determine the mass. After obtaining the mass of the individual single peptide by using the mass spectrophotometry, next again the foremost important step is determination of the amino acid sequence. This amino acid sequence is determined by using the database. So this is for example some of the amino acid sequence. This amino acid sequence is compared with the protein identified database. If the obtained unknown sequence is identified with the database protein sequences, finally we can identify the which type of the protein we have isolated. So there is a different techniques for the separation of the protein and identification. SDS phase and 2D dimensional gel electrophores. SDS phase is the technique used to identify the protein based on their molecular weight. For this purpose, we have to denature the protein by using the SDS, sodium dorsal sulfate, which give the uniform negative charge on the surface of the protein. So based on the unified charge on the proteins are migrated in a gel under the influence of the electrical field. Next, by separated the protein with the SDS phase, we have to run the 2D gel, two-dimensional gel electrophores. These electrophores separate the proteins based on their mass and based on their isoelectric pointing. Finally, proteins are separated in the individual protein peptides. So thank you. So this is the separation and identification of the protein by using the different proteomic tools.